Welcome to SCT 106 Biochemistry 1. In this session, we will explore proteins, under which we will study basic structure of amino acids, their different side chains or R groups, and the formation of peptide bonds between amino acids. We will also look at isoelectric point in acid-base titrations of amino acids, primary, secondary, tertiary, and quaternary structure, as well as the functions of proteins. Proteins are polymers of amino acids with each amino acid residue joined to its neighbor by a specific type of covalent bond called a peptide bond. The term residue reflects the loss of the elements of water when one amino acid is joined to another. Proteins can be broken down or hydrolyzed to their constituent amino acids by a variety of methods, and the earliest studies of proteins naturally focused on the free amino acids derived from them. There are 20 different amino acids found in proteins. All the amino acids have trivial or common names, in some cases derived from the source from which they were first isolated. Asparagine was first found in asparagus, and glutamate in wheat gluten. Tyrosine was first isolated from cheese, and so its name is derived from the Greek word tyros, meaning cheese. So was glycine from the Greek word glykos, which means sweet because of the sweet taste of glycine. In addition to these 20 amino acids, there are many less common ones. Some are residues modified after a protein has been synthesized. Others are amino acids present in living organisms, but not as constituents of proteins. Now, all 20 of the common amino acids are alpha amino acids. They have a carboxyl group and an amino group bonded to the same carbon atom. The alpha carbon that is shown in blue here, this central carbon here, is the alpha carbon, to which you have a carboxyl group bonded and an amino group also bonded to the same carbon. So this is where the name amino acid comes from. The amino group here being giving the part of the name amino, and the carboxyl group being a carboxylic group of carboxylic acids. So these two combined together give you the name amino acid. Now, these amino acids differ from each other in their side chains or R groups, which is shown here in red. The R shown here, the R groups or the side chains, are the part of the amino acids which differentiate these amino acids. And these R groups vary in structure, size, and electric charge, and they influence the solubility of amino acids in water. As we go on, we'll see the different side chains that are found in different amino acids. So let's first of all take the nonpolar aliphatic R groups. The R groups in this class of amino acids are nonpolar and hydrophobic. The side chains of alanine, valine, leucine, and isoleucine tend to cluster together with proteins, stabilizing protein structure by means of hydrophobic interactions. As you can see, glycine has the simplest structure among all amino acids, as its R group or side chain is a hydrogen atom. Although it is formally nonpolar, its very small side chain makes no real contribution to hydrophobic interactions. Methionine, one of the two sulfur-containing amino acids, has a non-polar thioether group in its side chain. So you see methionine having a sulfur in its side chain. There are only two amino acids with sulfur in them. We will be seeing the second one along the line. So methionine is one of those. So you see a thioether group, you see a sulfur bonded to an ethyl group and a methyl group. This is a thioether group that you have in methionine. Proline has an aliphatic side chain with a distinctive cyclic structure. So you see proline here having a distinctive cyclic structure in its side chain. Now the secondary amino group of proline residues is held in a rigid conformation that reduces the structural flexibility of polypeptide regions containing proline. So because of this distinctive cyclic side chain you have with proline, it is known to have this reduction in structural flexibility of polypeptide regions where they are found in those polypeptides. Now let's move on to aromatic R groups. Phenylalanine, tyrosine, and tryptophan with their aromatic side chains are relatively non-polar or hydrophobic. All of these can participate in hydrophobic interactions. The hydroxyl group of tyrosine can form hydrogen bonds. So you see the hydroxyl group of tyrosine here. The only difference between phenylalanine and tyrosine is the hydroxyl group on the aromatic chain. So you have phenylalanine not having a hydroxyl group, tyrosine having a hydroxyl group over here. And it is important, this hydroxyl group on tyrosine is an important functional group in some enzymes. Indeed, in some enzymes, this hydroxyl group acts in a way to hold substrates in place for the enzymes to act on them. Tyrosine and tryptophan are significantly more polar than phenylalanine because of the tyrosine hydroxyl group and the nitrogen of the tryptophan indole ring. So because of the hydroxyl group that you have in tyrosine and the nitrogen in the indole ring of tryptophan, these two amino acids tend to be more polar than phenylalanine. Tryptophan and tyrosine, and to a much lesser extent phenylalanine, absorb ultraviolet light, and these accounts for the characteristic strong absorbance of light by most proteins at a wavelength of 280 nanometers. And this is a useful property in the characterization of proteins. So you see the absorbance spectra of tryptophan and tyrosine here. You see the optimum absorbance of tryptophan and tyrosine occurring at a wavelength of 280 nanometers. These characteristic of aromatic amino acids give the property of absorbance of UV light of proteins, because these amino acids are very much contained in polypeptides or proteins. And so because they absorb light, proteins are found to absorb light as well at this wavelength of 280 nanometers. You have the optimum absorbance of UV light of proteins. Now the R groups of the polar uncharged amino acids are more soluble in water or more hydrophilic than those of the non-polar amino acids because they contain functional groups that form hydrogen bonds with water. This class of amino acids includes serine, threonine, cysteine, asparagine, and glutamine. So here we see the second amino acid with sulfur in it. This has a sulfur hydride group in its side chain. The first we saw was methionine, so the second is cysteine. The polarity of serine and threonine is contributed by their hydroxyl groups, that of cysteine by its sulfur hydride group, and that of asparagine and glutamine by their amide groups. So this polarity we talk about is due to the hydroxyl group here we find in serine, the hydroxyl group in threonine, the sulfur hydride group in cysteine, and these second amine groups which we find in the side chains of asparagine and glutamine. These groups are responsible for the polarity of these amino acids. Asparagine and glutamine are the amides of two other amino acids also found in proteins, and these are aspartate and glutamate, which are also referred to as aspartic acid and glutamic acid respectively, to which asparagine and glutamine are easily hydrolyzed by acid or base. Cysteine is readily oxidized to form a covalently linked dimeric amino acid called cysteine, in which two cysteine molecules or residues are joined by a disulfide bond. So you see cysteine here having the terminal sulfur hydride group in its side chain. A second cysteine can bond with this amino acid having a disulfide group. So you have one cysteine molecule losing the hydrogen on the sulfur hydride group, the second also losing hydrogen on the sulfur hydride group. So the two sulfur atoms in the side chain will then bond together to give the disulfide chain on the disulfide bridge, giving you a dimeric of this amino acid called cysteine. The disulfide linked residues are strongly hydrophobic or nonpolar, and these disulfide bonds play a special role in the structures of many proteins by forming covalent links between parts of a protein molecule or between two different polypeptide chains. The most hydrophilic R groups are those that are either positively or negatively charged. The amino acids in which the R groups have significant positive charge at neutral pH are lysine, which has a second primary amino group, and arginine, which has a positively charged guanidino group, and histidine, which has an imidazole group. So you see these amino acids having a second, you have lysine having a second amino group different from the first amino group which it has on the alpha carbon. You also have here arginine having the same positively charged amino group in this group which is called the guanidino group, coming from the similarity with guanidines, or guanine. Now you also have the histidine also having the nitrogen in its imidazole ring, also giving it that polarity. In histidine is the commonly amino acid, is the common amino acid having an ionizable side chain with a pKa near neutrality. In many enzyme catalyzed reactions, a histidine residue facilitates the reaction by serving as a proton or a proton donor or acceptor. So indeed, this hydrogen on the nitrogen of the imidazole ring is transferred in enzymatic reactions to either substrates or another amino acid residue in the active center of enzymes. And this transfer of proton is what activates some of these enzymes to act on their substrates. Now, there are two amino acids having R groups with a net negative charge at neutral pH, and these are aspartate and glutamate, also known as aspartic acid or glutamic acid, respectively, each of which have a second carboxyl group. So you see aspartate and glutamate having a second carboxyl group here, separate from the first carboxyl group, which is on the alpha carbon. You have a second carboxyl group here in aspartate. Here you have a second carboxyl group in glutamate. These second carboxyl groups is what gives the net charge of these amino acids to be negative. Now, the non-ionic form of amino acids does not occur in significant amounts in aqueous solutions. The zwitter ion predominates at neutral pH. So at neutral pH, you have the zwitter ion here on the right predominating in solutions rather than the non-ionic form. So you see the zwitter ion, which has a protonated amino group, and the carboxyl group here, which has donated a proton. This zwitter ion is what predominates in aqueous solutions of amino acids. When an amino acid is dissolved in water, it exists in solution as the dipolar ion or zwitter ion, coming from German. It's a German word. Zwitter ion is a German word meaning hybrid ion. A zwitter ion can act as either an acid or a proton donor, or a base, or which is a proton acceptor. So you see zwitter ion first of all here acting as a proton donor or an acid. So you see the protonated amine group releasing a proton, becoming neutral, NH2. So this is a zwitter ion acting as an acid, giving out hydrogen ions in solution. It can also act as a base or a proton acceptor, where you have the carboxyl group accepting a proton in solution and becoming neutral, and thus having a product that has a net charge of positive on it. So zwitter ion can act either as an acid or a base in solutions. Now, the substances having this dual nature are usually referred to as amphoteric or ampholites, coming from the word amphoteric electrolytes, because of the existence of both positive and negative charge on this chemical moiety, or this chemical substance. It's being referred to as amphoteric or ampholites, coming from the word amphoteric electrolytes. Simple monoamino monocarboxylic alpha amino acid is a diprotic acid when fully protonated. So you see a simple amino acid which is fully protonated, meaning the amine group is fully protonated, is carrying a positive charge. The carboxyl group is also fully protonated, is neutral, and the net charge of this fully protonated amino acid is positive. Now, this fully protonated amino acid could be donating a hydrogen ion from the carboxyl group and having the product being a zwitter ion having a net charge of 0. These can continue further to donate another hydrogen ion from the protonated amino group, becoming neutral, and thus the product giving you a net negative charge. Having gone through this, let's look at acid-base titrations, and specifically at the term isoelectric point in acid-base titrations. Now, acid-base titrations involve the gradual addition or removal of protons. Using glycine as an example, at a very low pH or in an acidic medium, the predominant ionic species of the amino acid is the fully protonated form. So in an acidic medium or at low pH, you have this fully protonated amino acid being predominant in solution. Now, in a titration where you have the first proton being donated to give you the negative charge on the carboxyl group, and thus giving you a zwitter ion or a neutral a species, an amino acid species that is having a net charge of 0 on it. When you are having this first stage which you see here, the first stage of the titration, somewhere in the middle point of this first stage of the titration, you have the concentrations of the fully protonated amino acid and the zwitter ion being in equimolar concentration at this midpoint of this first stage of titration. Now, as the titration proceeds further, where now you have the zwitter ion donating another hydrogen proton from the amine group to give you the negative species of amino acid in solution, because since you have the proton being donated from the protonated amine group becoming neutral and the carboxyl group remaining negative, the product you have is having a net charge of negative. Now, in the middle, somewhere in the middle, where you have all the positively charged amino acid fully donating the first proton, all the molecules in solution donating the first proton from the carboxyl group, and thus all of the species in solution are zwitter ions. And at this point where the release of the second proton is starting, at this point where you have all of these molecules of amino acids being the zwitter ion in solution, you have a net charge of 0 of amino acids in solution, a net charge of 0 of the amino acid solution. Now, the characteristic pH at which the net electric charge is 0 is called the isoelectric point or isoelectric pH, and it is designated with a symbol Pi. So this is what is termed as isoelectric point in acid-base titrations of proteins, or let me say of amino acids. Now, two amino acid molecules can be covalently joined through a substituted amide linkage termed a peptide bond to yield a dipeptide. So such a linkage is formed by removal of the elements of water or dehydration from the alpha carboxyl group of one amino acid and the alpha amino group of the other. So you see the hydroxyl group of the carboxyl group of one amino acid and the hydrogen of the amine group of another amino acid reacting to release water. This is a dehydration reaction or a condensation reaction. And as this happens, you have a peptide bond forming between the carbon of the carboxyl group and the nitrogen of the amine group of the second amino acid. Now, peptide bond formation is an example of a condensation reaction, and this is a common class of reactions in living cells. 3 amino acids can be joined by 2 peptide bonds to form a tripeptide. Similarly, amino acids can be linked to form tetrapeptides, pentapeptides, and so forth. When a few amino acids are joined in this fashion, the structure is called an oligopeptide. When many amino acids are joined, the product is called a polypeptide. Proteins may have thousands of amino acid residues. Although the terms protein and polypeptide are sometimes used interchangeably, molecules referred to as polypeptides generally have molecular weights below 10,000, and those called proteins have higher molecular weights. Now, for large macromolecules such as proteins, the tasks of describing and understanding structure are approached at several levels of complexity, arranged in a kind of conceptual hierarchy. 4 levels of protein structure are commonly defined. A description of all covalent bonds, mainly peptide bonds and disulfide bonds, linking amino acid residues in a polypeptide chain is its primary structure. And the most important element of the primary structure is the sequence of amino acid residues. So you see the primary structure here, where you have a certain sequence of amino acids in this peptide chain. This sequence of amino acids being lysine, lysine, glycine, glycine, leucine, valine, alanine, histidine. This specific sequence of amino acids in this peptide is the important element of the primary structure of this peptide. Secondary structure refers to particularly stable arrangements of amino acid residues, giving rise to recurring structural patterns. So you see the alpha helix being the secondary structure of proteins or peptides. Now, the holding of this peptide in a particular structural pattern, this arrangement, gives you the secondary structure in proteins, where you have some amino acids having some hydrogen bonding, or you have disulfide bond holding the structure in this alpha helix form, gives you the secondary structure of proteins. The tertiary structure describes all aspects of the 3-dimensional folding of a polypeptide. So this 3-dimensional folding of a polypeptide gives you the tertiary structure of proteins. And when a protein has 2 or more polypeptide subunits, so when you have 2 or more, you have one polypeptide subunit of this nature, and you have a second one, and sometimes third. When you have this protein with more 1, 2 or more polypeptide subunits, their arrangement in space is referred to as the quaternary structure of proteins. Now let's close the curtain on proteins by looking at some common functions of proteins. The first is the function of proteins as biological catalysts or enzymes for biochemical reactions, where they speed up biochemical reactions in the body by reducing the activation energy of reactions and by giving an alternate pathway to these reactions, for these reactions to occur timely for the sustenance of life. Proteins also are part of the structure of body tissues, such as the muscle, the nails, the hair, They can also serve as transporters across cell membranes where they form pores in these cell membranes. Or some of them, like for instance hemoglobin, which is a transporter of oxygen in the blood circulation system. Proteins can also act as neurotransmitters, where they serve in signal conduction in the nervous system. They also provide some immunologic defense in the form of immunoglobulins in the immune system. Then you have proteins also serving as hormones or regulatory molecules in the body. And lastly, some proteins can act as receptors on cell membranes for specific response. All of these functions of proteins, each of them can actually give rise to a discipline under biochemistry. So for instance, the function of proteins as enzymes can give rise to a discipline as enzymology. The function of proteins as part of the structure of body tissues give rise to structural biology. You have the function as neurotransmitters, giving a discipline in neurobiology, neuroscience. The provision of immunologic defense give rise to the discipline of immunology, and the function as hormones give rise to the discipline of endocrinology. So all of these functions of proteins, each of them could actually be giving rise to a separate discipline of its own in biochemistry. So this is what biochemistry reveals. Biochemistry is so broad and can actually give you the opportunity to choose in any of these fields in your further studies. On this note, I thank you for your kind attention and look forward to meeting you in our next section. Thank you.